Happy Monday. It's a pretty happy group for a Monday, I have to say. Especially considering how quiet this group is, right? You've already won the prize for my quiet group of BB350. Maybe that'll change, you never know. Okay. Well, I want to finish up saying a few things about peptide bonds and intersperse a few things about things that are related to amino acids. And then I'm going to talk more about proteins. And I don't think I'll get into hemoglobin today. But hemoglobin I'll probably talk about tomorrow. And I will warn you, I get very passionate about hemoglobin. And hemoglobin is probably my most popular lecture. If you look at my YouTube lectures, you'll see the hemoglobin is over the top in terms of numbers. So hopefully we'll have a good hemoglobin lecture this year. Okay. What I want to talk about right now are some more things about peptide bonds, which I started talking about last time. So let me bring you back. Well, actually, one more thing I just remembered. I forgot to give you a term, and it appeared in my highlights. I realized as I was writing the highlights that I didn't bring up something I usually bring up. And that's a term I think you should know because you'll hear it in other places. And that term is a zwitterion. Z-W-I-T-T-E-R-I-O-N. A zwitterion is a molecule whose positive and negative charges balance out and make it zero. Zwitterion comes from German, which I am told where it means it translates as hermaphrodite. Both positives and negatives. So it's got both sides. Okay? But anyway, zwitterion is a molecule that has equal amounts of positive and negative charges. So when we see an amino acid that has a +1 and a -1 charge, then overall its charge is 0. That's a zwitterion. Okay? All right. Well, as I said, last time I got started talking about peptide bonds and polypeptides. So I'll remind you that I said I used the term polypeptide and protein interchangeably in this class. Technically, there is a difference, but it's more subtle than we need to worry about in this class. Okay? You can see it's a polypeptide, and you can see on this figure that there's several things that are peptide bonds. Okay? So there's a peptide bond. That was originally a carboxyl group. This was originally an alpha amine group. And the two have joined together to make that peptide bond. We see several peptide bonds in here. And I pointed out how the peptide bonds are arranged so that the R group on one that's down is followed the next one by an R group that's up, et cetera. And they alternate in this way. And this alternation of R groups is an essential function or essential feature of proteins. You'll notice that proteins also have what they call direction. Well, the direction is defined by one end and another end, and those two ends will be important for you in calculation of charge, as we will see. Okay? So the first end is the amino end, and this amino end is the only alpha amino in the entire protein that's free. And it's the only alpha amino in the entire protein that can have a charge. All of the other alpha aminos get gobbled up in peptide bonds. So when you see a polypeptide chain and you see a free alpha amino end, that was the starting point of synthesis of that protein. Because proteins are made in the direction from alpha amino to alpha carboxyl. So the very first amino acid was actually this one on the far left. So remember that the only alpha amino that will have a charge is the very first one. All the others are gobbled up in peptide bonds. The other end of the protein chain has the only free alpha carboxyl. Because all of the other alpha carboxyls, again, got gobbled up in making peptide bonds. And importantly, that's the only alpha carboxyl that will have a charge. So now you see, if I were to ask you on an exam how to calculate the charge of a polypeptide, and I gave you a pH and I gave you the various pKa's for all of the groups, the way you would determine the charge of the polypeptide would be to determine the charge of the one alpha amino group the 1 alpha carboxyl group, and any R groups that can be charged. And of course, you know those as the carboxyl-containing R group amino acids and the amine-containing, or the basic R group amino acids. So that simplifies the calculation of charge of a polypeptide chain considerably. I'm not going to go through that here, but if you have questions or you want to see how one of those is done, give me a holler and I'll be happy to sit down with you. Okay? Okay. So that is what that looks like. One of the things that is interesting about a peptide bond is a phenomenon that's known as resonance. And resonance is a term that you may or may not have learned in organic chemistry, but resonance basically refers to the fact that electrons for a given bond are not in one fixed place. They can be in a couple of places. And this is particularly true for the peptide bond because we see that the electrons of this double bonded oxygen are not restricted to the double bonded oxygen, that in fact they can rearrange, as we see here on the right, and make for a double bond between the carbon and the nitrogen. So resonance means that these two structures basically swap back and forth. And it means that part of the time, the peptide bond has a double bonded nature to it. Part of the time, the peptide bond has a double bonded nature to it. Well, one of the things that you absolutely should have learned in organic chemistry is that when you have a double bond, you have cis and trans structures possible, right? Everybody remembers cis and trans. And a double bond creates a flat planar structure as opposed to a tetrahedral. Now that flat planar structure is depicted here as if that double bond that you see on the right were always present. Because since it's present some of the time, this structure behaves as if it's present all of the time. Now that will become important in a few minutes when I talk about Ramachandran plots, all right, which sound a lot worse than they really are. Okay, when I talk about Ramachandran plots. But Ramachandran plots are based on the idea that the peptide bond is at the center of a plane. The peptide bond is at the center of a planar structure. Okay. So resonance gives rise to a planar structure. Now, I want to just say a few words about some interesting related amino acids and peptides, and then I'm going to talk more about Ramachandran plots. So at this point, I've been talking about 20 natural amino acids that are found in proteins. And I've given you some examples of unusual amino acids that we see that are modified after they're in a protein. You saw that as an example of hydroxyproline or hydroxylysine. Okay? But another structure that, I'm just since another structure, but other things that are related to amino acids are listed here. So one of these is histamine. Okay? Histamine is related to the amino acid histidine, and it's involved in inflammation. And so when you have a cold, you understand that very well because your snot, okay, is being stimulated by the production of this molecule here, histamine. And that's why you take antihistamines to help reduce your congestion, because this is causing congestion, and antihistamine is reducing the production of this and reducing the phenomenon of congestion, that is inflammation and so forth. So that's what an antihistamine will do. And antihistamines, again, will stop the production of this molecule. This molecule is made from the amino acid histidine. Well, another, this is actually a dipeptide that we see that's produced. It's actually man-made, and you all know it, of course, as nutrasweet. Nutrasweet is an artificial sweetener, And it's an artificial sweetener that contains 2 amino acids, phenylalanine and aspartic acid. Structure's there. You don't need to know the structure. But suffice it, and there's actually a modification of it that's commonly done as well, but we won't worry about that modification. But it's commonly used, as you know, in diet soft drinks because it gives the impression of sweetness. And a lot of people think that this is actually a fairly innocuous artificial sweetener because, let's face it, it's made of 2 amino acids. And that's not quite the same as something that your body doesn't handle in some way. There may be problems with artificial sweeteners, and it's a very common question I get in this class. And there may even be a problem with this in very high doses. So I don't recommend going out and drinking 128 ounces of Diet Coke every day, which some people actually do. Okay? But in small doses, I certainly don't worry about this causing problems. Okay? Now, there are some people for whom this artificial sweetener will have problems, and these are people who are what are called phenylketonuria. They have phenylketonuria. So phenylketonuria is the inability to metabolize the amino acid phenylalanine. And if they get too much phenylalanine in their diet, what will happen is they will have some severe problems that will arise as a result of their inability to metabolize that. And I won't go through those here. But suffice it to say that a phenylketonuria should never have nutrasweet, and you'll see anything that contains nutrasweet will always contain a warning to people who have phenylketonuria. Phenylketonuria is a genetic disease, and it's a manageable genetic disease, meaning that once you are diagnosed with it and you watch your diet with respect to phenylalanine intake, you can usually not have any severe consequences. However, a lot of phenylalanine, as you might get in an artificial sweetener, can cause a real problem. Okay. Another interesting peptide that's found in our bodies, and I'm just going to mention it briefly here, I'll talk about it later in the term, is glutathione. Glutathione contains 3 amino acids, okay? And you can see them up here. You don't need to worry too much about that. They're glutamic acid, cysteine, and glycine, but again, you're not responsible for that. I only bring up glutathione to point out to you that it is a polypeptide. It's got 3 amino acids. It's very tiny. And our body uses it actually for protection against oxidation. It uses it for protection against oxidation. And we'll talk later in the term about the real damaging effects that can arise from oxidation inside of our cells. So this is one of our protectors against that excess oxidation that can happen inside of our cells. We have to have oxidation going on inside of our cells. I don't want to leave the impression that we don't, but what we're trying to control is uncontrolled oxidation. We need oxidation for energy, but those are all under tightly controlled circumstances. Uncontrolled oxidation is where the problem happens, and we don't want to have that. And glutathione helps to protect against that. Okay. And there's phenylketonuria. I'm not going to talk about oxytocin, but phenylketonuria. And this is the problem arising from phenylketonuria. People who have the inability to convert phenylalanine to tyrosine lack this enzyme right here. And as a byproduct of this inability to metabolize this to tyrosine, they get a bunch of side products made that are very detrimental and cause a lot of problems. Okay, questions about what I've said here? It's Monday, nobody's got any questions, right? Okay, well, let's turn our attention then to talking more about proteins in 3 dimensions. Okay, so far I've just talked about proteins from the perspective of the fact that they are polypeptides, 1 amino acid linked to another amino acid, linked to another amino acid. But I haven't said anything really about their structure. So now I want to spend some time talking about their structure. And believe me, there are entire courses, there are entire careers based on protein structure. So we're going to talk about protein structure in one lecture. You can imagine we're just going to talk a little bit about protein structure. Well, what you've learned about protein structure so far is one of 4 aspects of protein structure that people are concerned about. So far, I've talked about how one amino acid is linked to another amino acid is linked to another. And as you might imagine, the sequence of those amino acids makes a difference. Okay? The sequence, the order in which they are linked to each other makes a difference because each amino acid, because it has different R group, each amino acid will have its own characteristic chemistry. Consequently, it will have its own characteristic shape, and it will have its own characteristic activity. So if I change that sequence, I may very well change some of those components and result in something very different. So the sequence of amino acids is very important, and that's what we refer to as the primary structure of a protein. The primary structure is the sequence of amino acids. And I'm going to give you an answer to a question I frequently asked on a test right here, and that is, what protein structure gives rise to all of the other properties of a protein? The answer is the primary structure. The primary structure ultimately determines all of the other properties of a protein. So this tells you that primary structure is very important. It's so important that if we change that primary structure, we may very well change what the protein does, even if we make only a very slight change. Some slight changes will have very little effect. Some slight changes will have enormous effects. Okay. Well, primary structure is important because it defines that sequence of amino acids, and is what I hope to show you. That sequence of amino acids affects how they interact with each other. Amino acids aren't just laying out there doing nothing. They're chemicals. Chemicals interact. We've seen one really good interaction of chemicals so far, and that's been hydrogen bonds. I showed you how hydrogen bonds in water help give water a very high boiling point for its molecular weight. We're going to see now how hydrogen bonds give rise to a phenomenon known as secondary structure. So primary structure was the sequence of amino acids. Let me talk, first of all here, I'm going to come back and talk about phi psi angles in a minute, but let me talk first about alpha helix. What you see on the screen is a common structure that we find within proteins. This common structure is known as an alpha helix. I'll explain what you're seeing in a second, too. It looks a little weird, okay? And this structure is found in many, many, many proteins as a part of any of those proteins. As a part of any of those proteins. There are some proteins that have only alpha helix as structure. That is, they look helical. They're a wind and a wind and a wind and a wind. They go on forever like that, or seemingly forever. A really good example is your hair. Your hair has a protein that all it does is just make alpha helix after alpha helix after alpha helix on and on and on and on. Okay? Most proteins are not like that. Your hair is a little unusual in that respect. Okay? Now, what's happening here? Well, first of all, let me describe what you're seeing. What you see is a polypeptide chain. And that polypeptide chain, this green thing that you see, is not a part of the polypeptide chain. The green is there to guide your eyes, to show you the shape of this overall thing. The individual atoms are these little points and structures that you see there. Okay? So this is called a ribbon structure. The ribbon structure that's here is showing us what that helix actually looks like. Because if we don't have that guiding our eyes, there's so many atoms there we go, what the heck is that? Right? Now, I told you that hydrogen bonds give rise to secondary structure. And you can actually see the hydrogen bonds in this structure because the hydrogen bonds are labeled in yellow. The things that are yellow in this structure are hydrogen bonds between different atoms. Well, if you look very carefully, look at this. There's a hydrogen bond, 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 a hydrogen bond. And you see there's an abundance of hydrogen bonds within this helix. The hydrogen bonds are helping the helix to form and to stabilize it. The hydrogen bonds are allowing the helix to form and to stabilize it, meaning it will help hold it into that helical form. Everybody with me? How do hydrogen bonds form in a protein? Hydrogen bonds in a protein hydrogen bonds always have to have a hydrogen. The hydrogen comes from amines. The oxygen comes from a carbonyl group, and they interact. Okay? That's the hydrogen bonds that's happening inside of an alpha helix in a protein. Okay? Let's take another look at alpha helix. Okay? Anybody know where this is from? Does anybody know? Do you know? It's outside of Linus Pauling's house. Linus Pauling, of course, was born in Oregon. Linus Pauling went to Oregon State, got his bachelor's degree in chemistry in the 1920s. Linus Pauling was the discoverer of the alpha helix. It was the discovery of the alpha helix and another structure I'll show you in proteins for which Linus Pauling won the Nobel Prize. Okay? It was the first of his 2 Nobel Prizes. He won this one in the late 1940s. Okay? So in his front yard of the house that he grew up in is a structure of the alpha helix. Kind of cool, kind of nerdy, right? Okay. This is from, of course, we had the Linus Pauling exhibit in the library here at OSU. Many of you know that the Linus Pauling Institute, which was an institute that Linus Pauling started in his lifetime, is here at Oregon State. It's where the Linus Pauling Science Center is gotten its name from. And the Linus Pauling Institute, many of the professors from the LPI are in the Linus Pauling Science Center. And the OSU library has Linus Pauling's complete collection of papers. If you haven't seen it, I strongly recommend that you go and look at it. Okay? The Linus Pauling collection includes his Nobel Prize medals, which you can actually go and hold, solid gold. Okay? Pretty impressive, yeah. They've got them there in the library. They will let you hold on to them. Okay? They include all of his papers, including all of his correspondence with everybody from Einstein to, you name it, Watson and Crick, and so on and so forth. And this was Linus's actual notes when he was putting together and figuring out what an alpha helix was all about. Pretty cool. You can see the double-bonded oxygens, you can see the hydrogens, and you can see he's in the process of figuring out the interactions that give rise to that helical structure that we know as an alpha helix. A pretty remarkable piece of work. Okay. So the alpha helix is one kind of structure we find within proteins. Most proteins do not contain only alpha helix. I said our hair contains something called alpha keratin. Our hair is one example of something that contains pretty much only alpha helical structures. Right? But most things don't contain only alpha helix. Another structure that we see, another secondary structure that we see, is known as a beta strand. A beta strand. Okay? Sometimes you see this called a beta sheet. And it's a little confusing, so I'm going to try to clear that confusion for you. A beta strand, okay, is basically something that's oriented like what you see on the screen. It's an interaction between 2 parts of a protein, and this interaction between the 2 parts of a protein are again between a hydrogen of an amine and a double-bonded oxygen, but these are separated by some distance from each other. Okay? You could imagine that you see all these O's sticking out on the right, that those O's could interact with other H's, and you see these H's that could interact with other O's, that we could build a structure of 1, 2, 3, 4, 5, 20 of these together, And if we put 20 of these together, we would have a beta sheet. One of these is a beta strand. Okay? Now, beta strands are not helical. As you can see, looking at this, they are bent. They are like what we describe as the pleats of a curtain. They're just folds, are all they are. Okay? A helix has some roundness to it. These are point. That's the difference between a helix and a beta strand. Okay? So you might ask the question, well, when do you make helices and when do you make strands? And my answer to that would be, it depends on the primary structure. Certain amino acids will give rise to beta strands, and other amino acids will tend to give rise to alpha helices. No, I'm not going to hold you responsible for knowing which ones are which. Okay? But the sequence of amino acids will have an effect on which of those structures arises. You could imagine, based on what I've told you, that there are, based on the R groups being very different from one another, that some may pose constraints in terms of their size or their shape and prohibit one structure over another from happening. And you would be right. Okay? So we're not going to get into the details about which amino acids tend to prefer one versus the other. I will give an example in a minute of a couple of amino acids, but not related to alpha helices or beta strands. Most proteins will have some portion of them in an alpha helical arrangement and another portion of them in a beta strand or a beta sheet orientation. Most proteins will have a mix of the two. But these two structures are the most common secondary structures that we see in proteins, and again, they were both discovered by Linus Pauling. Okay. Now, the last thing I want to, let's see, right there's an example of a sheet, okay? Look at how the things are oriented. Here's one interacting with another, with another, with another, with another, with another. In some cases, they're arranged in the form of a barrel, where they're actually circular, and something happens in the middle. Yes? Two strands, two beta strands, yeah. Not a sheet. So, well, if you put two together, you're starting to make a sheet. That's why people sort of use the terms interchangeably. Technically, one of those is a strand, and as you start putting them together, at some point you call it a sheet. And there's not a specific place at which that happens. I'm a little unusual in distinguishing a strand from a sheet. Most people just say, oh, it's a beta sheet. You know. Okay. All right. Now, I want to say one more thing about structure here, and then I'm going to come back and talk about phi-psi angles. And that relates to turns. Okay? Turns. All right? If we look at this structure of this protein, and by the way, you see the ribbon structure again, and people tend to use ribbon structures again to sort of guide your eyes. And you see there's parts that aren't in ribbon structure, and that's because these parts don't have a regular repeating structure. So alpha helices have a regular repeat, that repeat being the helix going on and on. The beta strands have a repeat of a fold, 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 fold. Right? There are some amino acids that don't fit very well into those schemes, and they give rise to what we call turns. Okay? So here's a turn. We have we have an alpha helix, turn, another alpha helix, turn, another alpha helix, turn, etc. What you see is a specific protein shape, and that specific protein shape is a function not only of the alpha helical portions, but really importantly about those turns. Because the turns really are determining how the individual alpha helical components are arranged with respect to each other. These turns are really important for helping to determine what we'll talk about in a minute as the three-dimensional structure of a protein. I'm going to give you an example of an amino acid that is commonly found in terns. Proline. Proline, you remember, was the only amino acid I said whose R group came back and connected with the alpha amine. And I said there was less ability to rotate. There were more constraints on proline. And proline is less flexible than all of the amino acids because its R group is actually tied up with the alpha amine. As a consequence of this inflexibility, we frequently see, and this is not absolute, doesn't happen always, but we frequently see proline at the location of terns. Another amino acid we commonly see at the location of terns is actually glycine. Glycine's a little unusual. Why do we see glycine in terns? Anybody have any idea based on what I told you about glycine before? Yeah. It doesn't have an asymmetric center, and that means what? More flexible. Because it's got, so glycine, you remember, its R group is only a hydrogen. It's got the most flexibility of any amino acid. And so if you think about it being able to flex and make something into some orientation that you want, if you're doing Tinker Toys, anybody ever play Tinker Toys when they were kids? Tinker Toys give you some ability to rotate. The more ability to rotate you have, the more variety of structures you can make. And so glycine is also oftentimes found there. Okay. Now, I need to come back, or questions before I move forward? Yeah, question. Yeah. That's right. So you can see right there, there's a turn right there at the bottom. That's a turn, that's a turn. Excellent question, and absolutely. So when we think about the structure of a protein, turns really play a very critical role in determining that overall three-dimensional structure. Yes, absolutely. Okay. Now I said that there were some proteins that contain only alpha helices. Okay. There are some proteins that contain only beta sheets. Again, they're rare. One of them is silk. Silk contains primarily beta sheets. Silk has some pretty cool properties, as you've seen. It arises from structures that are primarily beta sheets. Okay. Well, let me spend just a little bit of time talking about this. Other questions before I move on? About phi-psi angles. Okay. I told you that the peptide bond had double bonded character as a result of resonance, which means that it behaves like a double bond. And if we think about it, here's one peptide bond in a polypeptide. Here's the next peptide bond in the polypeptide. And they've been drawn so as to show you that planar nature of these guys oriented here. In between the two is the alpha carbon. Now, if I'd like to show this as an example about thinking about protein structure. Now, we're not talking about alpha helical structures here because these are only at the level of 2 amino acids. Okay? Alpha helical structures arise every 7 or so amino acids. Okay? So I'm talking about between 2 amino acids. Here is one plane. In the middle of my hand is the peptide bond. Here is another plane of a peptide bond. All right? So I've got 2 peptide bonds here. And in the middle, I have an alpha carbon. The alpha carbon's my nose. Okay, everybody with me? Now, you can imagine that these 2 planes have places where they're not going to be very stable. They're not going to work because they bump into each other. And when you bump into each other at the atomic level, you have major problems. Okay? So how these planes arrange themselves will be a function partly of the R groups that are attached out here and partly of the angles between these bonds. Here's one angle, here's another angle. The angles are from the sticks that are my thumbs. Okay? Everybody with me? we can imagine that there are some rotational angles that will give rise to structures that bump into each other. Those angles are what we would call unstable or not allowed. Others will give rise to nice, regular structures. Structures that may ultimately be alpha helical in nature or ultimately beta sheets in nature if we go far enough. Okay? These two angles are what I call rotational angles, and it depends on how we rotate. So, as I go back here, I can think of this one on my left, your right, as 360 degrees if I could turn my hand all the way around, right? 360 degrees of rotation. This angle on my right, also 360 degrees of rotation. Some of those degrees of rotation are not going to give rise to stable structures. These rotational angles have Greek names, Greek letters, phi and psi. I'm sorry, phi down here and psi up here. You don't need to memorize which one's which, but you should know that phi and psi angles are rotational angles that help to orient the planes of a peptide bond. Now, those angles were studied by an Indian scientist named Ramachandran. Ramachandran was the first person to really recognize that there were going to be rotational angles that were going to be problematic, and he asked a computer program to predict, based on the known geometries of the atoms in a peptide bond, to predict which angles, would be stable and which angles would be unstable. Okay? He did the computer prediction for these, and it turned out that when people knew more about protein structure and they went back and they analyzed the known structures of proteins, that he was basically 100% right. Okay? The predictions of the most stable things in the geometry, okay, turned out to be the most stable, and the most unstable ones that he predicted turned out to be the most unstable. And it may surprise you to learn that there were angles that are in a Ramachandran plot. I don't have one up here to show you, and that's partly on purpose, because Ramachandran plots are a little confusing to understand. I have found that if I show them to students, they get all worried about, well, is this a minus 183 and a plus 37 over here, and how do they come together? And that doesn't really matter. The important thing about a Ramachandran plot is it tells you which angles are stable and which angles are unstable. That becomes very useful when you're trying to predict the structure of an unknown protein based on its sequence. Very useful. Okay? So Ramachandran plots are very useful and very helpful for that purpose. You are not going to interpret a Ramachandran plot. Everybody with me? People looking a little sleepy. You guys need a joke? A joke would do it? Does it wake everybody up? Okay, I'll tell you a joke, but here's the deal. I tell you a joke, you have to sing the song loudly today. Is that a deal? Oh, yeah, sure, until we get to the song, right? Yeah. Okay, this is one of my favorite jokes. I tell it in class all the time. I love this joke, okay? It's about Artie the Hitman. How many of you have heard my Artie the Hitman joke? One, but don't give away the punchline. Okay. All right. You'll get an F if you give away my punchline. I know who you are now. Okay. All right. So Artie the Hitman. All right. Artie is this little guy running around the neighborhood, right? And he decides he wants to start a career as a hitman. So he puts up signs all over the neighborhood, and it says, "We'll kill people for cheap." Okay. Puts his phone number on there, puts him on every telephone pole in the neighborhood, and goes home and waits for the phone to ring. And the phone rings. And this guy calls up and he says, "Is this Artie?" And he says, "Yeah, this is Artie. I hear you kill people for cheap." "Yeah." He said, "How much would you charge to kill somebody for me?" He said, "I'll do it for $1." "Wow. I'm just getting started, so I want to make sure I establish a name for myself, so I'm going to go do this for $1. "Okay. Who do you want me to kill?" And the guy says, "Well, I want you to kill my wife. So she's down at the supermarket right now. And if you could go," and he describes you to her, "and you can go and kill her." He says, "I'll pay you the $1. "Okay, sure." So Artie goes down to the supermarket, and he goes and he's looking around, and all of a sudden he sees this woman meeting the description just perfectly. Okay? And he says, this is my chance. So he goes up and he grabs a woman and he strangles her. Right there in the supermarket. He strangles her. He figures he's got it made, right? He turns around, he gets ready to leave, and he sees somebody saw him. Oh man, my career as a hitman isn't going to get very far if I get arrested, right? So he goes and he grabs them and he strangles them right there in the supermarket. Okay? Now he's getting ready to get out, and sure enough, a third person has seen him. He goes and he strangles them, and he figures, this is getting ridiculous, folks. So he goes running out of the supermarket as fast as he can. The police catch up with him. The headline in the newspaper the next day says, artichokes 3 for $1 at the grocery store. Oh. It kind of got you by surprise, didn't it? Yes. It always gets people by surprise. I think there's an element of surprise and humor. Artichokes 3 for $1 at the grocery store. Okay. Everybody awake now? What's that? You're going to sing loud today? All right, all right. Okay, let's talk some more about protein structure. I've talked about Ramachandran angles. I've talked actually about the next topic, which is fibrous proteins. Fibrous proteins are proteins that have only primary and secondary structure. They have only primary and secondary structure, meaning that alpha-keratin in my hair is a fibrous protein. I said it had that alpha helical structure that went on and on and on and on. Silk is a fibrous protein. It has only beta strands. I mentioned collagen the other day. Collagen is also a fibrous protein. Collagen, as I said, was the most abundant protein in your body. And collagen, I mentioned, was odd, because collagen is a fibrous protein. It has only secondary structure. And if we were to look at the sequence of collagen, what we would discover is it's full of proline that's been modified into hydroxyproline and glycine. Now, we thought about these with respect to turns, but now I'm telling you that here these guys can be arranged into a helix. Okay? So remember I said it was not absolute, that just because you had a proline that it was necessarily a turn. And it turns out that in the case of collagen, this is really important because the OH groups on this hydroxyproline, okay, can be joined together. So imagine I've got a helix of collagen over here. And I've got another helix of collagen over here. And it turns out that collagen helices are often bundled. How many people braid their hair? Anybody braids their hair? Okay, you braid. There you go. Okay. What happens if you don't put that rubber band on the end of your braid? It doesn't last very long, right? Well, with collagen, the equivalent of the rubber band are these OH groups that within the strands combine to split out water and make a covalent bond. So if we were to take your hair in that braided form and make covalent bonds between the strands, you wouldn't need that rubber band. Your braid would stay together. And consequently, that braided structure would be stronger, considerably stronger, than any of the individual hairs in your head. So everybody see how bundling gives rise to strength. Right? The bundling happens because the hydroxyl groups on these hydroxyprolines in collagen interact and give rise to covalent bonds. Well, why do I tell you that? Collagen, I said, was the glue that holds you together. Anybody remember what vitamin was necessary to make those hydroxyl groups? Vitamin C. We discovered years ago when sailors went off on long voyages, they didn't have refrigeration, they had basically salted meat that they put onto the ships that the sailors ate. They didn't exactly eat a lot of salad. They didn't exactly eat a lot of fruit. In fact, they ate none. They got no vitamin C. And these big, honking, ugly pirates would go out to sea, and they would come back as these wimpy little structures falling apart because their collagen was very weak, a condition known as scurvy. They developed scurvy because their collagen's falling apart. And because of those pirates having that problem, ultimately we did learn about what vitamin C does and how it makes for your body to stay together. Okay, so an interesting fibrous protein. Most proteins, however, are not fibrous. Most proteins are what we refer to as globular. Globular. What does globular mean? Well, I like to say it means that if you were to look at it, you would look at it and you would say, what's that glob of stuff sitting right there? Globular proteins do not have just one secondary structure. They have secondary structure, they have turn, they have other secondary structure, they have turn, they have other secondary structure, and all of these turns arrange this thing into a glob. Now that glob is not random. The glob is not random. It may look to your eyes like, whoa, okay? But in fact, that structure is very specific. It's very, very specific. And because it's very specific, that protein has a very specific action that it takes. It might be catalyzing a reaction. It might be carrying oxygen, as we'll talk about with hemoglobin next time. It might be giving structural integrity. It might be doing a variety of things. But its action or activity is going to be dependent upon that specific structure that it has, even if it looks to us like it's a glob. Here's hemoglobin. It looks pretty globby. Hemoglobin actually has 4 proteins in it. 2 identical units called alpha and 2 identical units called beta. We've just started talking about higher levels of protein structures. Let me back away from this and say something about the next level of protein structure. Oh, I forgot I had collagen up here. Sorry, here's collagen. So here's some of the bundling that happens in collagen that I was describing. There's your braid for you. And there's the sequence. Look at the glycines and prolines. And by the way, the HYP stands for hydroxyproline. Look at that in there. It's just abundant. But it can be twisted and turned into a helical structure. Okay. The next level of structure above secondary, not surprisingly, is called tertiary. Now, this is the last thought I'm going to give you today before we sing. Okay? And that thought is this. All right? Primary structure was a sequence of amino acids. Secondary structure was a regular repeating structure, and that regular repeat arose as a result of interactions between amino acids that were close to each other. I told you that the repeat of the alpha helix was about 7 amino acids. Less than 10 is close. Less than 10 is close. So these amino acids that are close to each other are interacting. You saw the hydrogen bonds on the screen for the alpha helix and beta strands as well. Okay? Those amino acids are within 10 of each other. That's what secondary structure is all about. Tertiary structure arises from interactions between amino acids that are more than 10 apart. More than 10 apart. Okay? This is the structure of a protein known as myoglobin. Myoglobin is related to hemoglobin, and we'll talk about both in a bit. But you can see that myoglobin has alpha helix, turn, alpha helix, turn, alpha helix. It's pretty much all alpha helix, and it has something else in here that we'll talk about next time. But you can imagine that here's one end of the protein up here where my pointer is, And the other end of the protein I don't even see. It's probably buried in here somewhere. But you might say up here at the very top where my pointer is, that's amino acid number 1. This might be amino acid number 20 down here. And you see that this amino acid is interacting with this amino acid over here. This might be amino acid number 100. This folded structure, and by the way, we describe these as folded proteins. This folded structure is arising because of interactions between amino acids that are far apart in primary sequence. Okay, so I'll talk about that next time. Questions about that before I turn us to singing? Okay, so I have a song about protein structure. It's called "O Little Protein Molecule." Oh, little protein molecule, you're lovely and serene. Can't hear you. With 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zing because of magic that occurs in protein folding. A folded enzyme's active and starts to catalyze when activators bind into its allosteric sites. Some other mechanisms control the enzyme rates by regulating synthesis and placement of phosphates. We'll talk about a lot of this later. And all the regulation that's found inside of cells reminds the students learning it a pathway straight from hell. So here's how to you like that, huh? Remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. See you guys tomorrow.